Welcome to another episode of the Dendrite podcast with Said and Nika. This is a conversation with Dr. Michele Ardolino. He's an immunologist and a senior scientist at the Ottawa Hospital Research Institute. He's also an associate professor in the Department of Biochemistry, Microbiology, and Immunology at the University of Ottawa.
During his scientific career, Dr. Ardolino has studied the mechanisms by which the immune system detects and eliminates cancer cells with a focus on natural killer cells and tumor immunosurveillance. His lab explores how cancer cells evade immune responses and how we can harness this knowledge to improve immunotherapies. His research contributes to our understanding of immune regulation in the tumor microenvironment with implications for cancer treatment and beyond. We hope you enjoy this episode, and here is Dr. Michele Ardolino.
Okay, good morning, Dr.
Good morning.
Ardolino. I'm very
Nice to
happy
meet
that
you
you're
guys.
here.
Likewise.
How was you had a trip earlier in the spring, right?
Did I?
I mean, you mentioned that we were trying to
Traveled. I went to many conferences. I wouldn't call them trips. The amount of
You're gonna be away for 3 weeks.
that was just meetings. It was just conferences. It's still very much fun. In fact, this is the part of my job that I like the most is when you go and
You get to
go
travel.
to conferences. Yeah, and the funny aspect of this is that when you're a trainee or you're a graduate student, you're very excited to go to see the science and learn and really get your wheels off and present your science. And I'll just go for drinking now. So I go there. And I take my gym shoes, and then I work out, and then I go to the bar. That's all my attendance to meeting. Most times. This is what I actually organized, so I had to be more, a bit more composed. But I would say that the networking is the important part now. But yeah, it was cool. It was fun.
Any particular bar or depending on the region?
It really depends where the meeting. So, for example, we went to Gatineau for the meeting that we organized, and there was no bar in the neighborhood. We went to the casino, the Lake Lamy, and the only bar was the one inside the hotel. So the choice was very limited, and there was a bunch of us. But whatever they would give us something to drink would be. Yeah.
You work with the options available.
Yes, precisely. Yeah.
So you've been around a lot of places, even in your just academic journey and in the institutions you have attended. What is your experience in in Italy and then in the US and in Canada? How did it all happen? It's a long journey, I know, but you can talk about it.
It's I liked a lot. I mean, the whole journey is really what made this career interesting. Being a graduate student for me happened almost by mistake. It was not very well thought, as many of my career decision was something just I thought this probably is gonna be fun, and so I just went for it. I knew I really wanted to study. It was the one thing that I was good at. Not very good at sports. Not very good at doing much with my hands, but I am very good at studying. At least I used to be when I was a student. And so I knew that I really wanted to be involved with something that would have me to read a lot. And so the way that I went to school. It's very common in Italy to just stick around the town that you are. So I was in Rome. I had a plethora of options for university. And at the time, I didn't realize how important it would have been to have a international background from the beginning. If I have one regret in my career, I think that probably is having not gone abroad for my doctoral studies. Although I landed in a fantastic lab. The place that I was in grad school was very supportive. They taught me a lot. My mentor was a very well-recognized scientist in the field. So it was very easy for me to find a job as a postdoc after in a very good lab. So I went to California after I graduated in Rome. I spent 6 years in Berkeley. And then after I did that, I came to Ottawa for my faculty position. And every place is different in many different ways. If you want to talk about research funding, that's one different. If you want to talk about the trainees, that's another difference. If you want to talk about the culture in the lab, that's it's very interesting. And being exposed to so many different things, I think helped me a lot growing as a person. Being young in Italy probably was good. It was a lot of fun. The university is surrounded by a lot of restaurants and bars and clubs. So what we used to do was to just, set up an experiment, and then we would just go out, have dinner, and then we'd come back and finish the experiment up. And the same in Berkeley. It was the campus is fantastic. You're surrounded by everything. So I would read I would leave my flow cytometry experiments up and running for hours, and I would be going out with friends and then come back and set up new plates. And here it's different because I'm older, so I don't have the luxury to have all this free time. And it works out well because there's not much around here, right? So there's just a limited option of things you can do. So you have to be in the lab.
A couple of parking lots. If you want to take
yeah, precisely. So that's what we do. But it's good, and I think the good thing of doing this profession is really, and the same for medicine, when you have to travel you have to move for your training from residency to fellowship, etc., is experience all the different options and changing quite dramatically often makes you a richer person. I think that was the part that I enjoyed the most.
I think like the nice part about it is that it's not that you're also living in that wherever you are. So it's not just a simple travel or just a conference. You're actually living there for a couple of years.
Yeah. And this is actually a good thing. I hear a lot of people now saying, I don't know if this is the right time to go to the States. And I still think that training in the States has a leverage, gives you a bit of a leverage. There's an advantage in just being in the States, particularly in some spots in the States, if you want to train in biomedical sciences. And what I tell trainees is that when you are there, but you're not really living there, right? You know that you're going to be gone. Mm-hmm. So it's a compromise that I think it was not hard for me to make. I knew I didn't want to live in the States, but I was happy that eventually I spent 6 years there and I experienced that reality, which is very different than here. It's very different than Europe. It's a very, very, despite, San Francisco, the San Francisco area being very progressive and very, there is a vibe, a European vibe, but it's really nothing like Rome or what Montreal would be. So.
And like your background, you're starting from Rome. It's quite difficult to live up to that.
Yeah, and I didn't speak any English. So if you think my English is bad now, you should have heard me 20 years ago when I went to the States. I was terrible. It was a very bad situation. I think I landed my job as a postdoc because my postdoc advisor is a lovely man, but he really, really, really likes to talk. So what I did was mostly listening and pretending to understand whatever he was saying. And I was like, I have no idea what you're talking about, but yes, I'm gonna nod. This is gonna be my strategy. And it worked out. I mean, eventually I picked up the English. I remember the one thing, the conversation that during my interview, he took me out for dinner, and the one conversation that we managed to establish on a more personal note was. He's a huge basketball fan, and I used to play basketball. I used to be a basketball referee when I was much younger, and so it was a connection that we managed to establish. And I knew a few words that I could tap into, and so that worked out for me. But if it was just a science thing, it would have been a disaster.
Imagine at the time you were a football fan.
Yeah, no, that would have been that would have. Yeah.
Why did you choose Ottawa after?
It was a number of reasons. I mean, when you are in the job market as a postdoc, so my biggest decision was really trying to decide whether to go to academia or being in industry. And at the time, so I am a cancer immunotherapist. My lab does cancer immunotherapy. So at the time, cancer immunotherapy was, quite newish. I mean, not that new, but there was still a lot of options, and still are, in fact, a lot of options for people who are being trained in that background to move to industry. But I didn't like the idea of being restricted to the kind of work that I wanted to do. So if you look at my lab now, we have 3 research axes and they have nothing to do with each other. And this is really what I really like because I never get bored. So we do evolutionary biology and then we do translational medicine. So from really nothing, just a computer and running alignments to trying to give patients some bispecifics that might cure them for their cancer. So we have a whole spectrum of research that we are digging into, And this is not possible in industry, at least not possible in industry at the level that I would have been able to do it as a scientist or whatever that position would have been. So it took me a bit of time. It was not much time, but it was a bit of time. So at that point, my career was like, okay, I'm going to be an academic. And then you have to be lucky. So you can have 7 papers in Nature, but if your qualifications and your expertise don't match the job search that the university is running, that they're not going to hire you. Because if they need a neuroscientist and I have 7 Nature publications on immunology, they're not going to hire me because I'm very good, right? They want somebody with that expertise. So I was lucky because there were many options that I have considered. There was many immunology positions when I was looking for jobs. And I knew that I didn't want to stay in the States for the reasons that we discussed earlier. I was lucky because my whole, tenure in the States was during the Obama administration, from day 1 to day whatever that I left. So that was good. But I knew that that's a crazy place. I don't know if, how stable it is. So I thought that I don't want to deal with that. And also there are a couple of things very sciencey related with the States that make me very uncomfortable. And so I thought that was not a good match for me. So I was looking into Europe and I got a quite nice fellowship to open up my lab at UCL, which is a, big school in London, UK. And there was a big pot of money. And I was, ready to sign. And I was very committed. I already have my wife. We were starting to look into jobs. We were looking into whether to leave, where to live, and I already had my mentors at the time at UCL were very happy to have me there, etc. And then I got a call from Ottawa saying that they wanted to recruit me to come for an interview. So I did come just because it's a free trip to Canada. Yeah. Might as well take it. Yeah, and I had a lot of fun. That time of your life you travel a lot because people want to hear what you have to say, so they take you out, they listen to your science, which is the best thing that a scientist can do, is to explain what they're doing. They take you out for dinner. At the time I didn't have any money, so that was good. And then they really convinced us that it was really a good fit. I thought it was a better fit than the one that London had to offer, because the money was not as good for the lab. The startup funds were not as substantial. So when you join a faculty, you are given some money from the university to run your lab. Precisely. You buy stuff, you pay students, you don't have to apply for grants, or at least you do, but you don't rely on the success for the grants to set up your lab. So you cannot do experiments. And the money that I would be getting in London would have been much more, substantially more. But I thought that the environment here was really better suited for me to succeed in many ways. One of the things that I really appreciated here was the richness of the department. The department here has a variety of expertise that it's very rare to find in other institutions. The North American structure of funding is something that I was more comfortable with, and the Canadian system resembles the US system in some ways, at least more than the UK system did. So I thought that I came here without having any real expectation to come, and then they convinced me, and they also actually did a very nice job trying to convince my wife to come. So they flew us over a second time, so not just me, but also my wife. Yeah, it was January 4 when we came here, so it was minus 27. I had to buy a jacket to be here because I was in California, right? So I had to buy a jacket.
That's a difficult start. Like, we convinced you.
But it was awesome because we were very scared of the cold. And then we came here for the coldest day of that year was that January 4th. And we were like, yeah, we'd survive. And I think in Insight it was the right choice because, we run a very balanced life here. It's much easier to live in Ottawa than London. London is crazy expensive. From the family point of view, this is a fantastic place to live. Raising children is much easier. And the university is a good university. Like, we do very good work here. Yeah, the faculty of medicine is awesome, and I'm lucky because I'm also, my lab is at UHI, as you know, so I can tap into both institutions quite heavily, and it's the best of two worlds. So I was very happy. Of course, London had fantastic infrastructures. The translational program was amazing. The mentors were really, really good mentors. So I'm very. It's a different story, but I'm very sad for those people. I always feel guilty for them because they invested so much in trying to get me there, and then I was like, "Oops, sorry."
You were hijacked.
Yeah, I was like, oh, "Sorry," but you know, that's science.
Where did immunology start?
Like, how did your passion with immunology come from?
I'm going to disappoint you here, but I really don't have a passion for immunology per se. I really wanted to. When I was in university, I took a bunch of different courses, and immunology was not my favorite one by any means. In fact, yeah, by many means. In fact, I started my training as an honor student in a biochemistry lab, but the graduate student who was working with me was not the most friendly person, I would say. And containing myself. And she was terrible. So my experience in the lab was terrible. I was like, okay, this is not something that I want to do. And then a position opened as a technician in a different lab, and I applied as a incoming master student. So I had to make some arrangement with my with my university director, the program director. And that was an immunology lab. And I was not like even in that lab, because that lab was also quite toxic. The environment was not the best, I thought. For a number of reasons. And I had so much fun, regardless, because the project was a bit enticing, and I had a chance of more getting my hands dirty with the experiments. And I was given a flow cytometer, and I was like, okay, play with that. And I learned how to do flow myself, which was very rewarding, because it's hard as if I give you a flow and tell you, learn. You will eventually figure it out. I guarantee you will, but it will take you a lot. And when you do, it's so satisfactory. It was like, oh, this is awesome. So I thought, I wanted to get out of that lab at that point for my graduate studies. And I thought, if I have so much fun in a immunology lab where the environment is not so cool, what it would be if actually I went to a immunology lab that, where people are nice, right? And they can teach me stuff. And so that's what happened. I went to the person who was running the program that would eventually ended up being my mentor. And she had one position open. I'll tell you why she gave me that position. It was not because I'm very, very bright. She came to the last day of class, and out of complete serendipitous, I baked a hazelnut cake, and I brought it to class. It was our last day of class. I thought, I'm going to be nice with my classmates. So she came, she had the cake. It was a good cake. I'm a very good cook. So she had a good cake, and she thought, okay, this guy probably, knows how to use his hands.
He's cooking. So.
Right? And he's nice enough that he's bringing cake for everybody. This was the first and last time that I brought cake to the lab.
That was a lucky day.
So it was really, but that's what I'm saying. You have to be lucky. Like in this job, this profession, you have to be very lucky. So that's something that you cannot, you have to build your luck, but at one point you have to be lucky. So that was my moment of, and so that's how I ended up in a immunology lab. And then it's fun, right? But I think, honestly, if I was in engineering, I would have been equally happy if I was given the opportunity to do research. That's what I really like, is the research, not so much the immunology.
At some point, you have some backgrounds to be able to understand a system, or at least be able to go deep in different parts of it.
Yeah.
And then, you just pick one, and you like what you become good at.
Yeah.
So that just progress.
But it's also very fun to try to challenge yourself constantly, and it's something that I always would encourage people to do, because if you get too comfortable in what you do, then it becomes, number one, boring. But also you lose that kind of cutting edge that you might have, because you're very good at something, but you can, unless you're really, really good at something, you can be good at something only for just this much time. and then you have to
That
move
gets
the target
outdated,
a little
yeah.
bit, because otherwise it's going to become stagnant. That's why I like in the lab to have some things that we have been doing for a long time and then we keep pushing, but also new avenues that we are exploring, because those are the ones that we can really bring our expertise in something that we were good at and now translate it into something else where we, didn't have that expertise, but we have the previous expertise, so we can kind of try to move the edge there a little bit. So I like this kind of approach. It never gets me bored, which is my number one concern. If I start being too bored, then it's not really
It's a time
worth
to move on.
Yeah.
Yeah. I mean, the money is not bad as a prof, I'll tell you that, but it's not as good that you want to be bored. That I need to be entertained. Otherwise, it's not worth my time. Yeah.
And what are the things that you do in the lab? Like you mentioned quite a lot of things. So
So what I do in the lab is nothing, because I have lost the ability of doing experiments. I think I'm still pretty good with mice, but I don't do experiments anymore. So most of my time is writing grants, and then mentoring students. So I know you have had me as an immunology teacher, but I guarantee you I'm a very good mentor. So if you were in my lab, you would see a different side of me, more nice and calm kind of and demeanor. So I spend a lot of time with my students. We have group meetings, we have one-on-one meetings, we discuss the science. That's the part that I enjoy the most. And then I do a lot of other less entertaining things for the university, including teaching. But the conceptual frame of my lab is we like to study the immune response. So the one thing that I really, really enjoy of the immune response is this idea of a homeostatic system that we discussed in class, right? So the fact that the immune system is a sensory system that detects changes and try to balance back and go back to where before the changes have occurred, or establish a new norm that is now the new physiological environment. And we started doing tumor immunology because, in my lab, as a postdoc, I was in a cancer immunology lab. I was becoming a cancer immunology lab, and so that was the interest. But also tumors are a very good model to study this, because there is a perturbation in the system when you introduce a tumor. Then you study how the immune system adapts to the tumor, and there could be two different avenues. One, the tumor is rejected, and so there is an immune response to reject the tumor, and that immune response now has to be controlled back. Or two, the tumor now becomes chronic, and that chronicity is something that the immune system needs to become used to. And there are mechanisms that happen in the immune cells that guarantee that the immune cells don't go awry. So it's something that intrigues me a lot. So we have a big part of my lab is studying the molecular mechanisms that make these killer immune cells adapting to the new tumor, to the new reality of having a tumor around. And this is also translationally important, because these cells in adapting become less responsive to the tumor, and so they are not longer able to kill the tumor cells very effectively. So if we identify what are the molecular switches that make the immune cells losing their edge in killing the tumor cells, then we can target those. And by targeting those, we hope that the immune cells will start working back again and become more responsive, and that's the concept of immunotherapy, basically. We also have a different part of my lab who's now became interested in this new, it's no longer much new, but this new for me at least, subset of immune cells, which are not great at killing, but they are really, really good at establishing inflammatory responses. So these are called ILC2s, innate lymphoid cells 2, type 2. And it's a great story on how we got into this, but for another time, maybe it's a lengthy story. But the idea is that these cells are very good at producing these inflammatory mediators. And we think that these inflammatory mediators, particularly in the ovaries, have an important role in regulating the physiology of the organs. And for example, for the ovaries, these inflammatory mediators might be very important into reproduction, right? Because you need to have an egg, and the ovulation is a very disruptive process. And so we need something that would fix the ovulation. We think that those cells are involved into, not fix the ovulation, fixing the tissues after the ovulation has occurred. we think that these immune cells are important in helping the tissue fixing. And so it's again that idea that there is a disruption to the homeostasis, which is the ovulation, then immune cells help bringing the system back to normal. And then we start looking into how these cells become important into cancer, in ovarian cancer and cancer immunotherapy. This is work that we are doing in collaboration with people at the HRI, including Bart van der Eyden, who is a fantastic ovarian cancer biologist. And you should have her on this podcast because she's a phenomenal speaker. So she and I co-supervise a student, or now a couple of students, to look into how ovarian cancer, if ovarian cancer can be treated by arming these cells with immunotherapy. And then we have a third angle in my lab where we are looking at this peculiar protein called PD-L1. I have to be a bit jargon here, which is thought to be the ligand of an important immune receptor called PD-1. It doesn't really matter. And we found that, and others have found that this protein has some very important cell intrinsic functions which were not thought to be the case. It was not thought to be the case. And this is interesting only to a certain degree. The thing that I'm really interested in now is that when we do science, there is a epistemological mindset, which is we name things, and in naming things, we attribute them a value. For example, if I tell you that this protein is a receptor and this other protein is a ligand, you are immediately inclined to think that the receptor is more important than the ligand because there is a redundancy in the ligands, right? You can have one or two or three, right? But you have a one receptor, and the one receptor is the one that does the action, right? So science happens very randomly. So we identify the receptors first, and then the ligands, and we know that the receptor has signaling capacity, but we didn't know that about the ligands. That's why the field has moved towards the idea that these proteins are receptors and these other proteins are ligands. But nature and evolution don't work that way. And what we in fact have found is that, contrary to what you would be inclined to think, the ligands are the one that came in evolution earlier than the receptors. So if you took a bunch of receptors that we're looking into, they showed up in evolution around, I don't remember the years, but around when, probably shark came up, which is a while ago.
Quite a long while ago.
There you go. But the receptors came out about 200 million years later, if I'm not mistaken. A number of millions of years later. So much, much longer. So those proteins that we call ligands and kind of dismiss them that way in some ways, they were there before their cognate receptor for a very long time. And so an evolution doesn't happen, the evolution doesn't keep proteins around if they don't have a function. And so those proteins have a function that doesn't really rely on the receptors. And that's why it's so cool for me, because the whole nomenclature of receptor ligand is kind of a misnomer if you think about it, because you're implying a hierarchical structure which is not there. And if for anything, the more important thing was there. And this also tells you that that's something about the immune system and how lymphocytes work, because these receptors are expressed mostly by lymphocytes, which is when these lymphocytes came around, two things could have happened. One, the environment adapted to whatever the lymphocytes were expressing. And so you have the receptor expressed by the lymphocytes, and then the, epithelial cells will start expressing the ligands, right? Because they have a bad interaction that is selected. But the other thing that could have happened is that the environment had all these proteins that were expressed, that now we call ligands, that they were having their function, they were fine. But then lymphocytes came around. And then when lymphocytes came around, the lymphocytes had to adapt to learn how to have those receptors that were able to bind to the ligands that were already present in the environment. And then the ligands and the receptors were selected in order to match each other. And this tells you how flexible the immune system is, and how it gives you a conceptual frame, I think, to how evolution have progressed to the development of lymphocytes, which is a disruptive event. It's something that really changed the way that we interact with pathogens, right? So it's very, very important. And I found this extremely fascinating. This is one of my favorite things that we're doing in the lab. And I have no expertise, so I'm very lucky I have a very good student on this. But, it's the breadth of research that we do is different enough that doesn't, again, get me bored, and so I'm always very stimulated. That's what I like.
So now I have two questions, because now thinking about like what is a development, I mean the stuff that we talked about in the class is the way that the cells are developing, like they are being modified heavily on the receptor, but the receptor is the one that's being like structured to see now what it would fit. So that it's like the ligand is the one, yes, the main one, and then you try to like work it out around that.
Yeah.
So it sort of makes sense.
One thing that I didn't tell you about is that the receptor that you're talking about, which is the T cell receptor, the B cell receptor, I think those would be the one exception where actually the ligand and the receptor co-evolved at the same time. These other ones are co-receptors, so they help the T cells and B cells to function independently. Well, no, they do depend on the T cell and the B cell receptor, but they're not the master regulator of their function. The development of the adaptive immune system revolves around these two proteins, like the T cell receptor and the B cell receptor. If you don't have those, you don't have much of those immune cells. So I think that would be the one exception. We're testing that as well, and there's more literature on that. But we're more focused on these co-receptors right now, but it's something that we're looking into as well. Yeah.
So what would have been, like, what was the function of the ligands before in those two million years?
Right. So we know the functions that these ligands have in mammals because it's very easy to study. So, for example, this one protein that we care about, we published this paper, the last paper that we published, about the way that it suppresses type 1 interferon. So it's a negative feedback loop. You have a viral infection, you have a lot of interferon that is produced. One of the genes that interferon brings up is this protein PD-L1. PD-L1 goes to the membrane and does two things. One, it suppresses lymphocytes because it binds to PD-1, and two, it suppresses the cell that is making interferon from producing interferon. That's what we discovered. So it's trying to close the loop to prevent that massive inflammation that interferon would do. And remember, we discussed this in class again, how the immune system is always very concerned in not having a out of control immune response. You always want to keep it balanced. So that's what one of the things that this protein does in mammals. But in like ants, ants won't have the right PD-L1, but in other lower, more ancient organisms, we don't know. And we don't have a way of testing it because the tools are not there. And we actually have discussed this at the last immunology meeting, the one that I helped organizing. We had a session on comparative immunology, which is basically what I'm telling you about, is these people studying ancient organisms. So there was a presentation about sharks, a presentation about sea urchin, etc. And the common denominator of those presentations were like, you guys with your mice, you have all the reagents in the world, we have nothing. And it's true, they have nothing. I don't know how they do work without all the richness of reagents that we have. It's amazing, and they do fantastic work, right? So we're trying to be clever, understanding what these proteins might do, but I think it's a very tall order for us. I don't think we're going to be able to really find it out. As my student always says, the best thing to do would be to take the easier thing to do would be to take a time machine and go back and see what it did.
But that is easier than like trying to find out what's...
Out of the three projects that you have at the moment, which one is like your favorite, that you're like the most intrigued about?
I guess the point is bouncing around with one to the other, like covers the whole.
But like, there's always one favorite.
I think it'd be easier for me to choose my favorite child. And it would be very easy for me to tell you the answer to that question.
Because I think you only have one.
I have two.
Oh, you have two?
I do.
Okay. We're introduced to one of them.
Yeah.
You have been, and that tells you a lot about that. Well, I'll tell you this. The more I write about something, the more passionate I become about something. So, but at the same time, the more I talk to a student about something, the more passionate I become about that something. So every time I have a student in my office talking about their project, I think, oh, this is so cool. This is the best thing we're doing. And then the next student comes up with a different project. I'm like, oh, this is amazing. This is what we should be doing all the time. So It depends. It really, I don't have a good, because they're all very close to my heart. I really, I wouldn't be able to pick one. It's a very difficult question to answer. Yeah. Different things excite me of the different projects. I think this one you're me talking about the evolutionary thing. I think this is so out of the box for us that intrigues me a lot. The ILC2 stuff has brought a lot of money to the lab, and so I am very excited. The NK cell stuff is more something that traditionally I've cared about for a very long time. So it's very close to my heart again. It's like, so yeah.
All in different ways.
Yeah.
I was wondering, are there like mathematical models on the immune system? Because
So people brighter than me have done fantastic work on modeling mathematically what the immune response look like. And they came up with this idea of quorum sensing, which is something that if you're interested in mathematics, you should definitely read. And it's beyond my ability to understand. But you know how quorum sensing works in bacteria, where you have a certain amount of, this is also recorded, so I don't want to say things that are not true. But my understanding of the quorum sensing is that you have a certain amount, for example, of nutrients that the bacteria as a population detects, and they have a coordinated response to that environmental cue, right? And T cells work in a similar fashion where you need a certain amount of antigen to...
Oh, it's all about the level of antigen.
Yeah, to mount that coordinated response. And people have mathematically modeled how much antigen you need and how much T cells you need to do certain things. It's very cool biology. There's actually one researcher in Canada, I think he's in Calgary, who does this, but it's not a lot of people doing it because it's very hard to fund. People don't get much money to do it. So, yeah.
Makes sense. What is like one of the most challenging things that you faced with like research or teaching or like in lab in general?
I think the most is center in my mind is quite sadly it's money, right? It's support in the lab. So most of my time, so my nights are spent just writing grants. Which is a very cool effort, except that the success rate is so low that, again, you need to be very lucky to be able to get a grant. And so that's probably the most challenging thing because we have so many ideas and, of course, if you talk to every faculty, they will have so many ideas and they probably are, all good ideas except mine, but, just a limited amount of money that can fund the system. It would not be too complicated for the government to increase this pocket of money. We don't give much money to biomedical research in Canada. Just to give you a comparison, it's this number that came up a few years ago that stunned me. But the biomedical research in Canada is funded mostly by this government agency called CIHR, the Canadian Institute for Health Research. And so CIHR has a certain budget, which I don't remember how much it is, but it's a budget, it's a pocket of money. This budget is less money than John Hopkins got from NIH.
It's one university.
Just one university. So the whole Canadian biomedical ecosystem relies on less money than John Hopkins does. And John Hopkins is not the exception, right? So that many other schools will have a similar amount of funding than the whole Canadian ecosystem. So that tells you a lot of what we can do as compared to the States and what the obstacles are. Sometimes there is a lot of frustration in researchers about the inability to really do what we could do if we had a better granting system. Because having not a lot of grants means not only that you risk to run your lab on a very tight budget, and many people do, including myself, but also you need to constantly spend time writing. And so at one point, it's good, writing grants is good because it's a good exercise, it's an intellectual exercise. But at the same time, it tampers with your ability to create new concepts because you're focused on one, but then you don't have much time to read outside of what you're doing. You don't have much time to experiment outside of what you're doing. So it becomes a very narrow activity at that point. And as much as I like it, I would like to have more time to do different stuff.
It consumes a big part of your focus and your energy.
It is. It does. Yeah. Yeah. It's so much that when I really need to write, I don't even budget it in my calendar. I just do it at night. It's just easier. If I have to fit it in my days, it would be too complicated. So I just decide, you know what, this month we're gonna write. So I got myself something to drink, and then I start writing. That's my strategy.
It's really complicated because my brother also had like a similar experience with studying for like masters here in the like U of T, and then for like his PhD, but then going to like Switzerland for the postdoc, and the amount of like the window of the stuff that you can do scientifically even speaking is much more different, simply because the amount of funds that are available. And then like it's the same case for US and I guess like other institutes. And
or even here, like, I mean, I got very lucky a few months ago. I was given money to do experiments, and even told to my trainees, they would tell you that my attitude towards just, let's do this experiment has changed dramatically from, before we got the grants and after we got the grants. Because I knew that, before we got the grants, there's just so many experiments we can do, and now after we can do way more. So if I have to be a bit more risky, I'm more inclined to do it now, because I know that, a high risk, high reward experiment might actually lead to a nice publication, more money to the lab, trainees having a better career, etc. And before, I would be a bit more adverse to the risk, because I'm thinking, okay, if I have to spend $10,000 on this experiment, it doesn't work. I don't have another $10,000 after this, right? I mean, it's over. This is all the chips that I have. I go all in on this, right? And that's a very stressful situation to have. That's one difference that actually between here and the States, that it's fundamental. To go back to your question of why I came to Canada, is in the States, your salary in many institutions is paid through your grants. So there is this concept of hard and soft salaries, where your hard salary is paid by the university and is a percentage of your salary, which can be as high as 75%. I think that's what the University of California pays, to 0%. So, for example, Scripps doesn't, to my knowledge, doesn't give you any money for your salary. So 100% of your salary has to be taken out from your grants.
There's also much more like personal interest as well involved
but
in
also
that.
stress, like the amount, and I would not be able to handle that. I mean, I need to have my paycheck every month, regardless, because I work, right? So I that's a non-negotiable. I do work every month. I need to get my paycheck, and so Canada works this way. So and then Europe works this way as well. You cannot take money from your grants to pay your salary, which is, I think, the right way of doing it.
This is so sad, because we also, like in my lab as well, like every time we want to do an experiment, especially if it's like a proof of concept and not an actual thing that we know is going to work, we have to budget in every single thing that we have to do, which is very frustrating, because we want to do the experiments, but we can't.
There's just so much money. I mean, the frustrating part is that, again, doubling the budget, there was this idea of doubling the CHR budget a few years ago. And if you look at the amount of money that it will take, is not that much money, and it will make our life much easier, because our success rate now is about 12 to 15% on one of these grants. If you double it, become from 20% to 30%. That's pretty good. Like, I can live with 25% success rate in my career. I would be
12% is so low. It's out of 10 grants that you write, one of them.
It's not even like that, unfortunately. So I'll show you my folder with all the grants that I've written and the ones that I've gotten. The ratio is not. I'm pretty lucky, but that's what I'm saying. You have to be lucky. So because you take a grant panel, many grants are gonna be very good grants, but you can only fund so many. So the panel that I got money from, there were, I think, 5 grants that were funded, and in the other panel 6 grants that were funded. So imagine how many other grants that were as good as mine didn't get funded, just because, it's luck. I got reviewers who liked my grants a bit better, and so there you go, here's the money. So that's the frustrating bit. There has to be some cutoff. You cannot give money to everybody. But the cutoff should be not as...
Did you see similar patterns in different, like even applying to med school? Like it's the same. Like I guess it's the whole structure of, I don't want to say bureaucracy, because it's easy to like get stereotypical on that, but I'm wondering if there are fundamental things that could change and could accelerate and expand many of these challenges.
I tell you what I think would change for med school is to not pay doctors much. Because in Italy, we have the same problem with med school being close access. Like you cannot just go to med school and say, hey, I want to be a doctor. You have to pass the test, right? But the numbers are way, way, way bigger. Like the acceptance rates is way higher than the one that you guys have here. Mm-hmm. But the reason why is that schools train these med students at a fraction of the cost that costs here to train students, and students don't get into that, and so they don't have to then earn so much money to get off that. So in Italy, you graduate from med school with, I don't think you need to accumulate any debt, to be entirely honest. Or if you do, like it's like, $10,000 or something like that. And so you catch up on that lost income very quickly as a doctor, right? But here you need to make, 200 grand a year to just try to pay off your med school debt, right? So, yeah, precisely. So it's like one of those things where you have to pay doctors so that they can get off debt so much. But then the system, we're paying the doctors, right? So just so many doctors you can train, because otherwise they,
big thoughts.
Okay, now switching gears a bit, you have used a lot of Bitmojis in our slides.
I do.
What is up with that?
I had a friend here when she introduced me to Bitmoji, and so when I was in Berkeley, I used to have a number of South Park characters. I don't know if you're familiar with the show.
Yeah.
So I did a South Park character for every lab member. I would be using those. And then I moved here, and there is this woman we friended, and she introduced us to the concept of Bitmoji in our group chats. And I thought, this actually is a Chrome extension. I'm just going to shove them in my presentation. So before I had Peanuts as well, Snoopy and something like this, but I thought the Bitmoji was more personal. And I don't like anything as much as I like myself. So if I can put myself in a slide, I'll do it. So it's, it keeps me entertained.
The one that I remember is like the one that's you on a couch in the beginning of the lecture, like, sit down. It's like, this is serious. We gotta talk about this now.
And if you notice, I have been too lazy to change the Bitmoji, but I have created the presentations over time with my hairstyle being very different and my dressing being very different. So I didn't make them consistent every time, but so you would see all the evolution of my hair over time. Yeah.
Do you have a particular, I'm talking about hair now. Do you have an interest, hair length?
It depends on the. So I have a problem with hair loss. So I have this alopecia disease, which is very interestingly, I find at least, the effect. So it's an immune reaction, right? So that's what happens to your immune system when it's not well controlled. So there's some lymphocytes in your follicles that attack the follicles, the molecule that drives this, one of the receptors that drives this, is the molecule that I studied in grad school, and is quite similar to what I was doing in grad school to some degree. So I found it to be quite entertaining. So
it's very personal.
Yeah, so it really depends on what my. So you guys didn't see it, but I had them pink one at one point, and that looked fantastic. I was very happy with the pink. So if I can manage to pull off a without old kind of head, I will do pink again, I think. I'll try this summer. We'll see how it goes.
Okay. Excited for that. So you also kind of hinted that you like research better, but you actually prefer research over teaching, right?
Yes, very much.
Why is that?
Well, there are a couple of things. So I actually like to teach a lot. The jokes on the side. I like this character of me not liking teaching, so that's part of it. But then I enjoy myself when I teach, and I like the interactions with the students. There are a couple of things that are really different. When I have just so many hours in a day, so I know that I can dedicate only so much time to do this or that. And as sad as it is, it's very, how do I say this? Let me regroup my thoughts. The simple answer is that most of my career is based on my research. So my promotions are almost exclusively based on my research. So if I got tenure, right, a few years ago, and I had, okay student evaluations, and I was teaching a decent amount, but what they really looked into was the research, right? So I know that for the university and the school, what and what really they're interested in expecting me to do is cutting edge research. Not so much the teaching is something that I have to contribute to do, but I can be less productive teaching wise and compensate with research. The research is for me more intriguing because there's always something new that comes up, and I know that you can do the same with teaching. Like, for example, Lisa does it, right? So Lisa and Keith are very focused on teaching, and they do that so greatly. I don't have the time and the intelligence to be able to do research and teaching at the same with the same magnitude of intensity. So for me, teaching most times is something that I have to do.
It's like a chore.
It is a bit of a chore. It's something that I have to do, but it takes away time that I could dedicate to do something else, like, writing a grant or do it. So I have to fit it in my schedule. That's the right thing. I have to fit it in my schedule. I have to fit the teaching in my schedule. And at the end of the day, I always think I need to get rid of this teaching. And then I always actually at the end of the semester, I think, I'm so glad I did it. Because I really enjoyed it after that, right? I don't enjoy preparing exams. I don't enjoy
Marking exams.
marking exams. I don't enjoy the complaints about the marking the exams. Sometimes students can be very disagreeable to deal with. And so whereas in my lab, for example, I'm surrounded by people that I really, really like. So if I have to decide to have a meeting with a student or a meeting with my students, I'd rather have a meeting with my students. So it's a personal thing. But I think if I were to lose all my funding and I would be needed to close my lab, I would have a lot of fun teaching, because I, at that point, would have all the time in the world, and I would dedicate myself to teaching. So at that point, I would, try to be more cutting edge with teaching strategy. Could I implement to do this or to that, or is there a way that students can be happier, more engaged, or can I try this multimodal thing? So it's a budget. So I have just so much time in the day.
And even the class, it's sort of like a...
You guys have felt this?
Not per se.
Well, I mean, one interesting thing, because, I people talk. And I talk to my brother because he's also, and he has, I see quite a lot of similar lines of interest at what I want to do and what type of student I like, what type of student I don't really like, and the type of exam. And he's like, yeah, I know that. So then you minimize the amount of time that you're spending on marking, which is the Sisyphus part of it. That it's just the homework.
Yeah. So the problem is that I really am for, as you've seen, the oral exam bit, right? So I like that very much. And I would only do that. If it was for me, that would be the only thing that we would be doing. But I think that would be... What do you think? Would that be a bad thing?
Well, my own personal...
Let's get rid of the midterms and just do the oral exam. That's what we do in Italy, by the way.
Just oral exams?
That was very interesting for the oral exam. I've never had an oral exam in university, so that was very new for me actually, and I quite enjoyed that.
Would you be happier?
Yes, I would think in person it's easier to explain the concepts, and there's no point of confusion, so you can get your point across. What is your favorite thing about Italy?
Gonna have to be food. I'm thinking what?
It could be a place, it could be
No,
food.
it's food. It's food. I'm just thinking what. Can I tell you what my favorite thing of Italy is? When you are in Italy, and this is true for Europe as well, many places in Europe at least, if you drive from point A to point B, you will find at least 5 restaurants that are worth going to. Mm-hmm. And I'm not talking about the strip mall restaurants, proper restaurants where you go and you stay there and you sit down and you eat. And if you're taking a road trip, you just can go to any little town. You go to the restaurant in that little town, you're gonna have the best meal of your life. It's super easy. Whereas here, if you go from here to Toronto, what restaurant do you go to, right? Yeah, right. That's the thing, right? So that to me is very distressing. Mm-hmm. That is something that I'm not used to, and I'm never gonna get used to. I saw this viral trend that somebody showed me about these American tourists going to Italy and stopping at we call them autogrill. I think you have them here in Ontario as well. These little stations across the road where they serve sandwiches, etc. And they think that they have the best food of their life. And this is what we consider very shitty food. Like in Italy, we never stop to the autogrill to get a sandwich, right? We would just go to the next door and,
I'm guessing it's also affordable. Like you can actually do that. You wouldn't go bankrupt.
That's true. Although salaries in Italy are way lower. So that is probably right now is the number one reason why we're not back in Rome, is because salary wise it would be very stark difference to the money I make now and my wife makes now to what we would be making back in Italy. So, but otherwise we'll be there. Yeah, I think so.
What is one city that you recommend us to travel?
Barcelona.
Okay.
It's funny that you don't mention Rome.
Yeah.
No, you should go to Rome if you have a chance. But I think Barcelona would be my favorite. It is my favorite city in the world. Still, I've been many times, and I would go back anytime I can.
Are you original from there or?
I'm from Rome. Yeah. But it's there is a beauty to Barcelona. It's on the sea. There's the mountain on the very back, so it's sandwiched between the sea and the mountain.
You also support the club.
I used to be kind of used to be one of my favorites. I don't care much anymore. I just lost my fantasy soccer championship yesterday for just half a point. So I'm still birds. So I actually enjoy the food there. The art is fantastic. There's actually not bad research going on there as well. So I sometimes snoop the website and see whether they're hiring in my field. Yeah, but the immunology is not the biggest thing. They have other area where they focus a lot, and
Again, it's always like the balance that you think that okay, if it's not big there, maybe like I could bring the babe, but again, it's like no.
Not that good.
And it's like the challenge of getting the funding, and like all of that stuff.
But the European Union Spain is part of the European Union when they have this European Research Council funds research at a very good level. You have to be very competitive to get one of those, but it's good money at that point. I like the ERC very much.
If you wasn't like being a scientist, what profession would have choose?
I like to think so. Now I would like to be a cook more than anything. I think working in a fancy restaurant would be very fun. I would like to be a chef. There's a lot creative. My wife will tell you that I experiment a lot in the kitchen. I'm the one that prepares all of our meals, and I'm very passionate about food. I like spending a lot of money. Go to restaurants. So it's something that I think I would see myself doing. I just don't have the training, unfortunately. So I miss the basis. I try to get better. I did a couple of masterclass, but yeah.
But that sets you apart from a lot of like the majority of people.
Yeah, I'm a very good cook. Like I remember the hazelnut cake episode. That was a good cake. I was like very young.
What's your favorite thing to make?
It comes and goes. So I started my cooking journey with pizza. I was terrible when I started making it, and now it's a good pizza. I'm trying to cook Korean. So I'm trying to nail down a couple of Korean dishes with mixed success. My biggest impediment is that the two kids don't tolerate much or any spicy food, or they don't have much fantasy, so they want to eat butter pasta or chicken strips. That's pretty much it. So if I go there and present them a kimchi fried rice, they would be like, there's no way again. So it takes a lot of time to do two separate menus, right? So sometimes I...
Do you make pasta yourself? Like, do you prepare the dough yourself?
Yeah. So that's actually my daughter knows how to do that. She's very excited when we have to do it. We try to do it on Sunday mornings. We make tagliatelle. And she's very happy when we have to do that. She knows how to make gnocchi as well. What else? Baste the turkey. If you have children, getting them, to help you in the kitchen is a very good bonding thing, and they learn how to use their hands a little bit. So it's they learn procedures, protocols. They know that they have to add this much sugar, otherwise, it's gonna not taste good. And so I think it's a very good teaching tool. That's why I also like in the.
And they will also, I think, if they collaborate, they will also eat the food.
No. So, for example, if I eat whatever I cook, even if it tastes disgusting, because I cooked it, so I was like, I'm gonna eat it now. It is what it is. But no, not my children. They would have a lot of fun making the food, and then they would just like, no, Still want my chicken strips. So
Remember the first pizza dough that I meant? It was like I was the only one, like I was the one responsible for that, so I consumed it myself. It was even not cooked, but it was the first one. But then I'm getting better. It's difficult with the house oven, like with that, but still.
it is. I'll tell you somebody a couple of tips later. Yeah.
Like setting the stone there to like warm up and
Yeah, that's one. Use a polish. Do you use a pre-fermented dough or use straight into the?
what I do is like depending on like what I'm trying to make, like try to rise and then like I don't beat it. I still want it to be like fermented, so it's like airy, so it becomes like crispy.
Okay.
And then
I like to ferment in two batches. So I do the pre-fermentation, then it's called, I don't know the English for this. I think it's called Polish, but it might be wrong. It's called Biga in Italian as well. There's two different flavors of it. But and then you do the second round of fermentation, and that makes it very, very airy, and the gluten structure becomes super tight. So that's that. You can do the flipping in the air.
I've never been able to do that.
You have to do 2 steps. Yeah, you have to do 2 steps, but it's a lot of fun. Then the children love it. They have a lot of fun looking at that.
And what would be your favorite topping? Like do you
Toppings?
do additional like margarita or like
We usually have when we do pizza night in my house, we usually bake like, I usually bake like six pizzas at the very least, because I know that my wife and my children would like to stay more on the safe side, so we'll have a couple of margaritas and maybe a pepperoni slide. Then we go a bit more experimental.
exotic?
Yeah, no pine nuts.
I was about to ask, like, is it that disrespectful?
No pine nuts, sorry, pineapple.
Is it that disrespectful to have like breaking the pasta or like putting pineapples on the pizza?
It's not disrespectful. It just tastes very badly.
He likes it actually.
Doesn't mean anything.
I wasn't like I was completely against it, and until
I
I
don't
tried
know.
it was like it's like I don't know, having apples and cucumbers at the same time. Like it's two completely different things. They taste different, but like if it's available,
I think if you, that's the thing. I think food is culture. So if you do something with a certain mindset, everything is good. If you're just doing it in the American way, what I would like to say,
just add cheese.
then that is not the way. But if you, for example, take a piece of pineapple, thank you very much, and you char it on a barbecue, potentially you can even smoke it if you're really good at it, and then you caramelize it, and then you add some more toppings that
Go well with that.
establish the flavor of the and so at that point it becomes something gourmet, and then I'm all for it. So I would have no hesitation saying that. But if it's a slice of pineapple on it, that is something that I'm not happy to live with. So that's done without culture. So I have no tolerance for that.
There's no background. It's just an abomination. Do you have any favorite restaurants here?
I like Wales Bones quite a lot. It's on Elgin. It's in front of the Lord Elgin. It's not the best restaurant in Ottawa, I think, but it's my favorite. It's so when we have faculty come over, we always take them out to dinner.
That's like the go-to.
Yeah, it's a very nice. It's a bar kind of vibe, but the cuisine is quite sophisticated, but it's more pub fare like. So the oysters are fantastic. The oysters to die for, and they have very good beer. They have this thing where the oysters, you can eat them with toppings, etc. And so they give you a little flask of whiskey that you put on top of the oysters, and that's very, very cool. I like that very much. So I like Wilspoon. Yeah, I would suggest Wilspoon. Even without the whiskey and the oysters, you can still enjoy it.
And is there any like pizza place that you approve of?
Quite many. We I think where did we go recently? We went to this place called it was on Preston, of course, Del Piacere, and it was very authentic. Like we had a lot of fun. The kids loved it. We like the pizza from Anthony. It's quite good. We like the pizza from Roberto. It's quite good. We try to go to pizza places that are family oriented, so that the children can make a lot of noise. What I find to be a little bit concerning is the inter-visit variation between the pizza that you have in any place. So if I go to Anthony, I can be very lucky in one day get the best pizza, like, oh wow, this is like being in Rome. And then the next day you go back and it's like doughy and it's, it's very plastic-like, it's chewy. And that happens many, many times. So we go to, I like Romolo, for example. And then we go sometimes and the pizza is great. Sometimes it's like, it's hit and miss. So the lack of consistency is what distresses me the most. So that's why I like to do pizza myself as well. It's also cheaper.
It's a surprise, like every time, you don't know. So it adds excitement.
It's an expensive surprise. Like if we go the four of us, it's like $150 for the four of us to have pizza. Yeah, and you compare it to back home where you spend, I don't know, maybe €50 for four people to, maybe not under 50, but €60, something like that. I don't know. It's very cheap.
Every single time, like it.
It's good. It's good pizza. Yeah, it's very hard to get bad pizza. Yeah.
Where do you call home right now?
It's here. Yeah, it's here. Yeah, we voted, so if I can vote, this is my home. Yeah.
Would you still travel back and forth?
We are very happily because mine and my wife's parents are still alive, so we like to have children seeing them. We have family there. Our siblings are all there, so we don't want to, but at the end of the day, we always go every year. So, for example, this summer we are not planning on going. Then yesterday we're like, okay, how much is it gonna cost us? So we kind of resigned that we'll have to go. So.
I mean, they also miss their grandchildren and children. So.
Yeah. So we kind of, when they're gonna be dead, it's gonna be easier not to go, I think. For the time being, we're going. Yeah, it's good that they don't speak English, otherwise this would be a disconcerting topic.
Do your children, do
they speak Italian? Yeah. They have. We always speak Italian to them. It would not make sense because both my wife and I are Italian. So I would.
Do you remember the first day of class? You mentioned that you're speaking English after 1 month, and it's like it's
yes. Yeah, so that's the problem when you start, and it's good that someone does the first part of the teaching because I get a bit of time to get back to my yeah.
Do you do any like sports even now?
I run.
Okay.
I go for runs.
Do you do marathon?
I got new shoes yesterday. No, but the longest I ran was 20 kilometers, but was out of the context of a marathon, of a half marathon. I just end up running a lot. It was a good day, but I ran 10K. That's my go-to kind of thing. Very slowly. So, but I think as a scientist, running makes a lot of sense because it gives you time to think.
And I wanted to ask that, when you run, do you listen to something or do you just thinking?
It depends. When I go to run outside, I listen to music, or I used to have a podcast that I really liked, and now it's over, so I don't listen to that anymore. But and when I run on the treadmill, I usually watch a movie or watch a seminar. It's very sad, but that's the reality of it. But what happens is that the endorphins and everything then have a clear head after. And even if I'm stuck on writing something, I find myself very often go for a run, come back, and then everything is now fluent. So it's a subconscious kind of thing. I don't even think too much when I run. I really don't. But then something happens in your brain that works the magic itself. Now I feel bad if I don't run, so I really have to go. Yeah. So I'm going today for a fun. I have a fitness class that I'm going to. So my 11:30 appointment is my wife and I. My wife works upstairs, and we go together on Tuesday to this fitness class that the gym offers here. So it's a good thing. It's not running, but I'm probably gonna run later.
And now the ultimate difficult question.
Yes.
We always save for the last. Yes. What is the meaning of life?
That is a question that has shifted a lot for me, and I think that it is a moving target. I think when I was young, my development was my meaning of life. I wanted to be. I mean, you always want to be happy, right? But I wanted to be the happy version of myself, and I worked a lot in trying to accomplish that professionally in a way that was very, as everything that I do, kind of serendipitous and, unwavy, if you will. So it was without a real scope. But I knew that if I would be doing, and I hoped that if I would be doing something that I enjoyed, then everything would have worked out. Because I have a bit of confidence in myself. So I thought, you know what, I'm going to study it and something is going to happen good for me. Because if you study, that's you're going to be good, right? So, and then, in my personal life, this was not as straightforward, I would think. And now I think that the focus has shifted a little bit, both personally and professionally, towards not just myself, but the people that you interact with. So I like to think that, on the personal side, what I will contribute to the world is to, I have 2 children, that they would be good people, right? They would not be like this. Yeah, be good people, right? So that I think is the meaning of my personal life, is to, being able to contribute to the world with 2 people that would make the world a little bit better. From the professional standpoint, I very quickly realized that your biggest contribution to science is not the discoveries that you make, because, there's just so much that you can do and do well, right? So I had a decent paper that I published, and that was what I think an okay contribution to the field. But, how many people are going to read it, right? But the training of the people that you do, that's really what's going to last. Because I, in a career, can train dozens of people. And those dozens of people will eventually end up training more people. And then so
Becomes a chain reaction.
Precisely. To me, the biggest thing for me has always been the pursuit of knowledge. So I realized that I can only pursue so much knowledge-wise. And training people is the ultimate way of trying to get to know as much as you can, not just for yourself, but, just attack this huge block of unknown and chew it up from different angles, right? So that I think is what I'm shifting more and more. My emphasis is, even it's easier because now I'm a bit more established, is really what can I do for people that I train, right? So how can they be the best version of themselves? How can they be progressing their careers, etc.? So I think your next person is where you should find your meaning, because it's ultimately the most rewarding thing. I've been very lucky with my career, and I found myself to be quite happy when I get a grant or when I get a paper published for maybe 3 seconds. It's super happy, but it lasts 3 seconds. It's gone. So the time that we celebrate, I'm already, whatever, we do another thing. But then I look back at, the couple of students that I trained, and I have this sweet spot this sweet feeling in my heart that really fills me with joy every time that I think about them. Like, every time that and even students that I'm training right now, like I know that some of them are on the way to go, and I feel like I feel so melancholy about them, right? And I think it will be the same with my children when they will leave. I would like to see them prospering and being very happy, and I think that would make me ultimately very happy. Good question.
Well,
And that's
it was
a beautiful
pleasure.
answer.
Yeah,
Yeah.
it was a pleasure to have you.
Thank you very much.
Both in class and in the conversation. Thank you very much.
Thank you very much.
Thank you.
Thank you for listening to this conversation with Dr. Michele Ardolino. If you're interested about his work, you can find the link to his website in the description.
This is a Dendrite podcast. To support it, please share this episode with your friends and anyone else who might be interested. We hope you enjoyed this episode, and see you next time.